Hello, everyone, and welcome to today's webinar, The ABCs of Biosafety Cabinets. I'm Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Labconco. To learn more, visit labconco.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Jeremy Sandler, Product Manager at Labconco. Jeremy brings over 20 years of research experience in the life sciences to Labconco as the Product Manager for Biosafety and Laminar Flow. His research spans microbial ecology, plant ecology, genetics, genomics, stem cells, and tissue regeneration. Jeremy is passionate about developing the most advanced and comfortable biosafety equipment for tomorrow's scientists. Jeremy, you may now begin your presentation.
Thank you, Cassie. Us today, I will be talking about biosafety cabinets. In this talk, I like to call the ABCs of biosafety cabinets, and you'll find out why. So a quick agenda today will be defining biohazards and assessing biosafety requirements for your labs. We'll be talking about Class 2 biosafety cabinets, the most common in labs, explaining their subtypes and how they're used, and then exploring how to pair a biosafety cabinet with your application for your lab. So let's get right into it. What is a biohazard? It's a good question. A biohazard is any infectious agent or a part of it that presents a possible hazard to the health of animals, humans, or plants. This could be bacteria, viruses, other pathogens, and we classify biohazards in several ways. Those classifications can be found with several resources. And I always like to say the first resource and best resource that you would have when thinking about biohazards and doing work in the lab with biohazards is your institution's biosafety officer or environmental health and safety office. These are experts on working with biohazards, containment, and safe handling practices, and they are really your go-to people on campus or your institution. But there are also several other resources out there. The CDC in the United States produces the Biosafety and Microbial and Biomedical Laboratory book. This is almost like the Bible of biosafety. I have one on my desk right now, and it includes information on every possible hazardous pathogen you could imagine, viruses, bacteria, others, how to use them safely, how to handle them, and the types of containment they would need. Canada and the World Health Organization published similar guides. And then each biohazard would be divided into its risk group, and there are specific guidances based on the risk groups of the biohazards. So let's get into those. Risk groups assess the relative risk of a microorganism to humans, plants, or animals, and they take into account the pathogenicity, how serious the infection could be, the transmission, the types, or how easily transmitted it is, the availability of preventative measures or treatments like vaccines or antibiotics. And taking all of these things into account, biohazards are divided into 4 risk groups, 1 through 4. And there are 4 corresponding biosafety levels that are used to contain biohazards in these different risk groups. These are BSL 1 through BSL 4, or biosafety level 1 through 4. And you're probably familiar with these terms from working in biosafety level labs. Either, usually BSL 1 or BSL 2 are pretty common in academia and other research settings. So BSL 1 labs, the lowest level of biosafety level, these are labs and environments that work with biohazards that actually don't cause disease in healthy humans. This would be common research organisms like E. coli, Bacillus subtilis, yeast, things like that they're not hazardous or pathogenic at all. These biohazards, these pathogens, can be used on open bench microbiology. So what you would think of as a typical laboratory bench setting. And really the only guidelines are that these experiments in BSL-1 labs or with these microbes should take place in a dedicated laboratory space with its own sink. and there really aren't a lot of serious precautions for containment or isolation of these microbes. Moving up the scale to BSL-2, this is when we introduce agents that can cause mild diseases in humans or are difficult to spread by aerosols. These are things maybe like clinical samples, cell culture, pathogens for diseases that aren't very serious, but still could cause disease in humans. And in this case, we need to increase the safety and containment and the personal protective equipment that researchers would use. So when working with these pathogens in a BSL-2 lab, users would employ a biosafety cabinet, class 1 or class 2, and we'll talk about protective equipment based on their pathogen or the agent they are working with. So on the top, you would see researchers in a BSL-2 lab, maybe working with non-pathogenic cell culture. These are cells that could be cultured from humans or animals. And in this case, the containment is actually probably more for the protection of the samples and the cells than the protection of the researchers, since these cells probably would not get them sick. On the bottom, though, you see a researcher with much more serious PPE. He could be working with clinical samples or samples of viruses or bacteria that could cause illness, and that's why he has a higher level of personal protective equipment on. BSL-3, this is working with pathogens that could cause severe or fatal diseases in humans, but we have treatments. At first, when COVID was first being researched, it was a pretty serious virus, and COVID was initially classified as work done only in the BSL-3. Now that we have a vaccine and treatments exist for COVID, it's actually been downgraded to BSL-2. So there's an example of a real-world pathogen, and it's how it's used in different biosafety labs. So there are lots of safety measures added on to a BSL-3 lab. That includes a corridor where researchers would have to shower in and shower out, self-closing doors, enhanced PPE, so you can see that this researcher now has a full face mask and respirator on, and a negative pressure lab exhaust, so air in the lab can never leave the lab. Finally, a BSL-4, this is a pretty high-level biosafety lab. This is really only for diseases and pathogens that cause fatal disease in humans with no vaccines or treatments. So think of the most serious hemorrhagic fevers, Ebola, Marburg virus, things like that. BSL-4 labs are really rare, and we're not going to be talking a lot about them today. Either a researcher would have to use a positive pressure suit with a biosafety cabinet, or a class 3 biosafety cabinet, which is completely sealed without their positive pressure suit. So now that we've covered some of the basics of biohazards and the different levels of biohazards, let's talk about containment enclosures and safety enclosures in the lab. So when I say a lab hood, you might be thinking of several types of laboratory equipment or pieces or instruments. The most basic and common is a chemical fume hood. This is a negative pressure box where room air is drawn in through the face, over the work surface, and then out the top of the fume hood through ducting that's connected outside of the lab. There are no HEPA filters, and there is no personnel protection. This is great for chemical vapors, but actually not an ideal place to conduct cell biology research. That's because anything that's already in the lab on the outside of the fume hood goes directly over the sample. So let's say somebody sneezes or coughs, bacteria or viruses could be expelled, and those would land directly on the samples on the work surface of the chemical fume hood. So not an ideal place for conducting cell biology research. The first thing we think of with a safety enclosure for cell biology is a clean bench. This uses a fan to pass air through a HEPA filter up above, and then the HEPA filtered air flows over the work area. This provides a sterile, clean environment for cell culture, but the problem is anything on the inside of the work surface of the work environment comes directly out onto the user. So this would not be used in any type of class 2 or 3 biosafety application where the user would not want to be exposed to any pathogens on the inside. If we reverse the fan from a clean bench, we get what's called a Class 1 biosafety cabinet. In this case, it's almost like a fume hood with a HEPA filter for the exhaust. That means unfiltered lab air flows into the work area. It passes over the experiments and then is exhausted through the HEPA filter and cleaned and sterilized before it's exhausted or recirculated into the lab. This would be okay for working with pathogens, but you should still be aware that anything on the inside is not protected from contamination from the rest of the lab. So this is still not ideal for cell culture, where the user would want to keep the cells sterile and clean and contained. And finally, we get to a Class 2 biosafety cabinet. This uses both negative and positive pressure and dual HEPA filters to contain the air inside the work area through a HEPA filter, and then exhaust through a separate HEPA filter. So this means that whatever is on the inside of the work area stays on the inside, whatever is on the outside stays on the outside, and it only passes through HEPA filters before going in or out of the biosafety cabinet. And here are some examples of different laminar flow or safety enclosures. On the left is a horizontal clean bench, and you'll see that large screen on the back wall. That's actually a HEPA filter behind that diffuser. These are used in pharmacy compounding, sensitive electronics, manufacturing, things like that. A vertical clean bench is almost the exact same as a horizontal clean bench, except the HEPA filter is on top instead of on the back wall. And this just provides a slightly different airflow in case of large equipment being on the inside. And then finally on the right is a Class 1 biosafety cabinet, also called a powder containment hood or a containment ventilated enclosure, depending on the setting. This is where the HEPA filter is on the top, air is drawn inside from the lab and then exhausted through the HEPA filter out to the environment. These are all great enclosures for limited applications, but today we're going to be talking mostly about Class 2 biosafety cabinets. So when you take concepts from a clean bench on the left and a Class 1 biosafety cabinet in the middle, you get a Class 2 biosafety cabinet on the right, where there is complete containment of whatever is inside the work area and separation through HEPA filters, so no biohazards from the inside would ever escape and no contamination from the lab would ever get to the inside of the work area. Air from the lab is drawn in through the front grill and directed below and behind the work area, and it only passes over the work area after it's gone through the HEPA filter for the supply air, or it's only exhausted into the lab after it's gone through the HEPA filter for the exhaust on the top of the cabinet. And here is an illustration or a short video of laminar flow in a biosafety cabinet. We'll give it a second to load. Does not look like the video is loading. Oh, there we go, finally loaded. So you can see we have a smoke generator at the top of the biosafety cabinet, and you can see the one-way airflow from the top down to the bottom. And you can see either the laminar air is captured through the rear airfoil slots and then moved through the HEPA filters again, or through the front grill and then taken beneath the work surface and recycled through the HEPA filters, the same pathway. This is important because it's a one-way laminar airflow. There's no mixing or swirling of air. That means we always have a clean curtain of air over the entire work surface. What are the parts of a Class 2 biosafety cabinet? First of all, the purpose is to provide an enclosure for the safe handling of biohazardous materials like bacteria and viruses. And Class 2 biosafety cabinets provide personnel protection. That means they keep the user safe. They provide sample protection. That means they keep whatever is on the inside safe from contamination from the outside. And they also provide environmental or lab protection, which means they don't let biohazards from the inside recirculate through the lab. There are lots of different applications for Class 2 biosafety cabinets. These include cell and tissue culture found in lots of academic labs, microbiology, pathology and clinical diagnostics, assessing clinical samples and diagnostics from patient biopsies or swabs, pharma and biotech. So a lot of pharma and biotech companies use biosafety cabinets themselves when they're working with cells or producing some of their products. And also pharmacy compounding. Let's say a hospital has to mix up a sterile IV of medication to be injected into a patient. That could actually be mixed and compounded inside a biosafety cabinet to maintain sterility and protect the pharmacy technician from possible hazardous drugs. There are several really important parts of a Class 2 biosafety cabinet, and they all contribute to the safety and containment. We'll start with the inlet grill. That is the row of holes you see at the front of the biosafety cabinet. This is an area of high negative pressure, which both captures any air approaching from the outside lab and also captures downflow air once it passes over the work area and touches any biohazardous agents. So the inlet grill captures all air that could cross the barrier. The work surface itself, both the flat work surface and the walls, are all made of stainless steel. This makes it really easy to clean and disinfect. Stainless steel is great for wiping down with 70% alcohol or other sporicidal agents. You should always note, though, that some agents that can contain bleach could corrode stainless steel. So we always recommend wiping down with 70% alcohol after using anything that contains bleach, just to make sure there's no residue left over and to prevent corrosion of the stainless steel. The glass sash is a really important safety device. Unlike a fume hood, where the lower the sash is, the safer the fume hood operates, in a biosafety cabinet, the sash needs to be used at a very set specific height. And this is because the balance of inflow air through the grill and downflow air from the top HEPA filter are calibrated to the height of the sash. If the sash is too low, it will constrict the inflow air and actually force it over the grill into the work area and could contaminate samples on the inside. If the sash is too high, the inflow is insufficient and contaminated biohazardous air could come out into the user's face. So with a biosafety cabinet, there are actually alarms that will tell a user if the sash is out of place and needs to be restored to its correct operating height. And then up above in the plenum is where all of the air handling happens. And this is what the plenum looks like. On the right side is the blower, which powers the entire biosafety cabinet. The plenum in the center is an air distribution center. It distributes air either through the supply HEPA filter on the bottom or through the exhaust HEPA filter on the top. And this is really the heart and lungs of the biosafety cabinet. There are several types of Class 2 biosafety cabinets, and we'll start with a type A2. This is the most common. Probably about 80% of all biosafety cabinets sold today are type A2s, and that's because they are the most efficient and the easiest to operate. All types of Class 2 biosafety cabinets provide the same personnel and product protection, but they work a little bit differently. So a Class 2 type A2, as you see on the right, actually recirculates most of the air through the HEPA filter and work area many times. As the air passes through the HEPA filter and then over the work area, it's captured either through the rear or the front airfoil, taken through the back channels, through the blower again, and then through the HEPA filter, and a smaller portion is exhausted out of the biosafety cabinet. This exhaust is important because the exhaust makes sure that there is always negative pressure in the inlet grill to contain biohazards. One drawback of a type A2, though, is that it can never be used with chemicals. And that's because any volatile chemicals on the inside could, if they spill, could create either a high percent atmosphere on the inside of the biosafety cabinet, or they would pass through the HEPA filters into the lab. So while HEPA filters are great for containing biohazards, they actually don't contain chemical vapors. And that's why we can never use chemical vapors with a type A2 biosafety cabinet that recirculates into the lab. This can be addressed, though, by adding a canopy. So a canopy is a way to connect a type A2 biosafety cabinet to a building exhaust system. This way, all of the exhausted air, instead of going back to the lab, goes outside of the building. There's still recirculation, so you can't use a large volume of hazardous or volatile chemicals because you could still get a vapor buildup inside the work area, but you could use smaller amounts of chemicals in the work area without concern. And you should always consult your institution's biosafety officers or EH&S office just to make sure they are aware of chemical use in biosafety cabinets and you have the correct setup. So about 40 years ago, type B biosafety cabinets were developed. These are kind of dual biosafety cabinets. The work surface is partially exhausted through the back, but is partially recirculated through the front, just like a type A2. All of the exhaust air is drawn out of the biosafety cabinet and out of the building through ductwork and a fan on the roof. And the work surface has this kind of unique feature. We call this the smoke split because if you have a smoke generator over the biosafety cabinet, you can see that smoke either goes towards the back and is exhausted or goes towards the front and is recirculated. That means in the rear of the biosafety cabinet, you can use hazardous chemicals because that air is exhausted. In the front of the biosafety cabinet, you cannot use volatile chemicals because that air recirculates and could build up hazardous vapors. One problem, though, is that it's really hard to determine where the smoke split is on the work area. And it actually changes. As the HEPA filter loads over the years, the smoke split can move. As the weather changes, the temperature outside or the atmospheric pressure changes, the smoke split actually moves because the exhaust fan behaves slightly differently. Type B1s are not the ideal biosafety cabinets to use for this reason. It's not really obviously clear where to use volatile chemicals and where it's not safe, and where it is safe and where it is not safe. So a type B2 biosafety cabinet answers that, and a type B2 is completely exhausted. There is no recirculation within the work area. This means there is a smaller blower which pulls in air from the lab, passes it through a HEPA filter, and over the work area in a single pass. The exhaust is powered completely by a fan on the exterior of the building, and that fan is responsible for exhausting the entire work area and providing the negative pressure in the inlet grill for containment. One downside, though, is that if the building exhaust fan would ever fail, the supply fan needs to immediately shut off. And that's because if the exhaust fan fails and the supply fan keeps running, the work area could become positively pressurized, and that means that biohazards would come out the front of the biosafety cabinet directly at the user. For this reason, NSF, the certifying agency in the United States for biosafety cabinets, has determined that if there is ever an exhaust failure, the system must shut down its supply fan within 15 seconds to try and minimize any breach of containment. Because there's a single pass airflow and it's completely exhausted, this requires a lot more blower power to drive the exhaust of a type B2 biosafety cabinet. There are also some considerations with the type B2 biosafety cabinet. So NSF, NSF 649, which is the gold standard for biosafety cabinet certification, says that each B2 should have its own dedicated exhaust blower. CETA, the Controlled Atmosphere Testing Association, Controlled Environment Testing Association, which is a group of manufacturers and certifiers of biosafety cabinets, says each type B must have its own dedicated blower. And I'll show you why we really recommend and say that a type B must have its own dedicated blower. So let's say you have two type B2 biosafety cabinets with one blower, and they are joined with a duct connection. Initially, the inflow for both of these is set to 105 feet per minute, which is standard. Let's say over time in this lab, the users prefer the biosafety cabinet on the left. Maybe it's next to the window with a nice view. Maybe it's a bit more comfortable. For whatever reason, they prefer the one on the left. That means over the years, it's going to be used more, and those filters will load faster than the filters on the right. The same blower is connected to both biosafety cabinets. If the biosafety cabinet on the left with the higher use, the filter loads faster, there's going to be more resistance to pull exhaust, and that means the inflow is going to be lower. On the right, the other way, the opposite occurs, where there's less use, the HEPA filter loads slower, and that means there is less resistance on this HEPA filter. The blower can pull harder through this biosafety cabinet. And this means on the left, with a lower inflow, there could be a possible breach of containment where biohazards leave the biosafety cabinet and come into the lab. And on the right, this means that there could be a lack of containment from the lab inside, where contaminated air could rush over the inflow and then touch the work surface before it can be filtered. In this case, we would recommend separate valves that can be adjusted on each B2 biosafety cabinet just to make sure they can each have their own dedicated and precisely controlled airflow. But there's a solution to the type B problems, and that's a type C1 biosafety cabinet. And I like to call these a hybrid biosafety cabinet. That's because there is a portion of the work area in red here in the center which is completely exhausted, and there are portions of the work area that are recirculating. And the type C1 can actually switch back and forth. It can operate in A mode, which is like a type A2, where the entire work surface is recirculating. Or it can operate like a type B biosafety cabinet, where the center is completely exhausted outside the building. And that means that users can actually have volatile chemicals in the center while it's exhausted. And the type C1 also has a really interesting safety feature, and that is a second blower. So here's kind of a cutaway of a type C1 biosafety cabinet. You can see the blue blower on the right side provides the downflow that would cover the entire work area, and also the recirculating exhaust on the sides. But the red blower on the left exhausts the center work area through a separate channel, and that would go outside the building. Unlike a type B2, which has no exhaust fan and relies completely on the building exhaust, this type C1 has its own exhaust fan in red, and that means if the building exhaust ever fails, this exhaust fan can keep running for several minutes to provide safe time for the users to close any chemicals and close any biohazards before there's any problems with containment. So a type C1 really is a flexible hybrid biosafety cabinet that provides the ultimate safety containment backup. When we look at the exhaust, so CFM on the top row is the amount of air flowing through the biosafety cabinet for the exhaust. And static pressure, you can think of how hard the blower has to work to move the air. So think of a straw in a drink. If you're drinking water through a straw, it's pretty easy to suck that up. But if you're drinking a really thick milkshake, it's harder. So that's the static pressure. A type B2 has a much higher static pressure because there is no exhaust fan to help move the air. But a type A2, when it's connected to the canopy exhaust, or a type B2, when it's connected to its own exhaust, has a much lower static pressure and a much lower CFM because the unit itself helps move the air. And that translates into a direct savings for labs. Laboratory air is expensive. It has to be conditioned correctly before it enters a lab for the correct temperature to make sure the researchers and the experiments can be conducted safely. And it also usually is a single pass through the building. So it's pretty expensive to run a building's exhaust system for a lab. A type B2 connected to the building's exhaust over the 15-year lifespan costs almost $100,000 to run, and most of that is energy cost. A type A2 with a canopy is about half the cost, $45,000, and a type C1 with a canopy, very similar to a type A2, still around $46,000, $47,000. But in this case, a type C1 and B mode, you can use any volume of volatile chemicals. So you get the same chemical ventilation as of type B2 with the energy usage of a type A2. So this means a type C1 really is a flexible, versatile biosafety cabinet for many situations. And here's a chart you can look at that would just give you an idea of the ideal usage and chemical safety for the different types of biosafety cabinets. And you can see that the type C1 all the way on the right literally checks all the boxes for biosafety cabinet operation. So now that we've discussed different types of biosafety cabinets and how they're used, how they can be used with chemicals, let's talk about applications in the lab and what types of labs they're used in. So going back to the different BSL labs. BSL1 labs, most of this microbiology work is conducted on the lab bench, and there is no biosafety cabinet needed. It's up to the user if they want to have a biosafety cabinet. Let's say they want to keep their cells sterile, they don't want to contaminate them. That would be a good case for a class 2 biosafety cabinet, even though there's no biohazards to be taken into account. BSL2 labs, most commonly cell culture, but also working with diseases like hepatitis, salmonella, Lyme disease, HIV. Even though HIV is a very serious disease, because it cannot be spread by aerosol, it's classified as a risk group 2 and can be used in the BSL2 lab. In all these situations, a class 2 biosafety cabinet is the right choice. It provides the protection the user needs to keep themselves safe from the biohazards, and also make sure that the research conducted on the inside is sterile and not contaminated from the rest of the lab. BSL3 labs. So in these labs, again, researchers use biohazards that could be fatal or very serious, but we have treatments. So this would be things like anthrax, tuberculosis, West Nile, COVID was used in the BSL3 lab. Now that we have treatments and it's under control, it can be used in the BSL2 lab in smaller volumes. Malaria, swine flu, these would all use class 2 biosafety cabinets. Again, they provide excellent containment for the biohazards on the inside, and they make sure that the research taking place on the inside is sterile. There are a few other interesting applications. So at the beginning, I touched on a class 1 biosafety cabinet. This is what I like to call, or used in what I like to call messy science. This is things like grinding or powder milling of non-hazardous items, maybe microbiology or molecular biology experiments that generate aerosols. So you have a centrifuge on the inside, you're homogenizing tissue, anything that's messy and that you want to be contained, but you don't care about sterility. Because remember, all the air that's drawn into the class 1 biosafety cabinet comes directly from the lab without being filtered. And I mentioned centrifuges. I should also note that centrifuges should never be used in class 2 biosafety cabinets. That's because centrifuges usually put off a plume of warm air and exhaust it pretty quickly outside the back of the centrifuge. And this can actually disrupt the laminar airflow inside the class 2 biosafety cabinet. Another safety precaution for a class 2 biosafety cabinet is never using open flames. Let's say open flames for a Bunsen burner for sterilizing a loop for picking cells. Those open flames, also the heat plume disrupts the laminar flow and can actually damage the HEPA filter and cause serious containment issues. So centrifuges and open flames are two things that should never be used in a class 2 biosafety cabinet. Class 1 biosafety cabinets are also used in mail sensors for opening suspicious packages. They contain whatever is on the inside, but we don't really necessarily care about having a sterile environment. These are, though, somewhat uncommon in academic and government research labs, and that's because most of the things that are done in the class 1 biosafety fume hood, with the exception of biohazards. So if there's a true biohazard, it should be done in the class 1 biosafety cabinet. But let's say you're doing non-hazardous tissue homogenization, that can be also done in the chemical fume hood. Compounding pharmacies. So I mentioned these earlier. Compounding pharmacies prepare drugs for patients on the spot. Class 1 biosafety cabinets in compounding pharmacies are called containment ventilated enclosures, or CVEs. They're used with working with hazardous drugs. So they provide an environment with negative pressure where anything on the inside is passed through a HEPA filter before a leak going back into the lab. So they protect the user in the lab. But these could be drugs that are not injected. So maybe creams or ointments that are spread on the skin, things that don't need to be completely sterile, but are still hazardous to the person preparing them. These are all done in a Class 1 biosafety cabinet. Class 2 biosafety cabinets are used with sterile compounding. So the best example probably is chemotherapy. You think of a cancer patient who probably has a very compromised immune system from either the cancer itself or from chemotherapy. Chemotherapy drugs are very hazardous, especially in highly concentrated forms. So on the right here, you see probably a pharmacy technician preparing chemotherapy, where they have a concentrated drug vial and they are diluting it very, very precisely into a sterile IV bag for the patient. The patient must have a sterile drug because it will be injected directly into their body, and the user must be protected from the hazardous drug on the inside. So a Class 2 biosafety cabinet is an excellent choice for sterile compounding of hazardous drugs. Lab animal research is also a really common use of biosafety cabinets. Animal procedures can be done inside the Class 2 biosafety cabinet. Anesthesia, injections, surgery, cage changing or weighing and measuring. These are all things that are done especially in immunology research. So immunology or cancer research, a lot of these mice or other lab animals have a very compromised immune system. So we need to provide a sterile environment on the inside to keep the animals healthy and make sure the experiment is conducted in the most clean, rigorous way possible, and also protecting the user from any possible hazards from the animals. And then lastly are large containment enclosures for robotics or liquid handlers. These are used more in pharma, biotech, or preclinical research labs for conducting cell screening or drug screening on different types of cells. They could be used in clinical diagnostic labs for pipetting and arraying clinical samples in large format. Cell sorters or flow cytometers could also be used if there are biohazardous cells being sorted. And also other types of liquid instruments or lab automation instruments. All these instruments are too large for a traditional biosafety cabinet. So we've developed these large Class 2 enclosures that are much bigger and can hold all of these robots and more. One thing I should note is that while these large containment enclosures meet the NSF requirements for containment, they are not NSF listed. That's just because the NSF has not yet adopted the standard for larger containment enclosures. So with that, I think I'll wrap up with the slides I had, and I'm happy to take any questions you have.
Thank you for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Q&A box below and click submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question is asking, how often do HEPA filters need to be changed?
That's a great question. So it really depends on the type of work that's done inside the biosafety cabinet and where the biosafety cabinet is located. Let's say you have a biosafety cabinet inside a clean room in a pharmaceutical company, and they are doing filling and finishing on vials. This is a very clean environment, very few particulates. These HEPA filters could last 15 to 20 years. In a typical lab, though, with cell culture and multiple researchers using the biosafety cabinets, still we would say about 10 to 15 years between HEPA filter changes. The biosafety cabinets that Labconco makes actually do a live filter monitoring constantly during operation. So we have a torque sensor and an RPM sensor in the blower, and as the filters load over time, the motor has to turn harder and work harder to maintain the same airflow. So we actually measure how hard the motor is working to move air through the HEPA filter. We know the maximum RPM, and we actually display 0 to 100% of filter life remaining based on how much more RPM is available in the blower. So generally, though, to answer the question, in a standard cell biology lab, 10 to 15 years is a good number for how often HEPA filters should be changed.
Excellent, thank you. All right, our next question here. This one is asking, what is the difference between HEPA and ULPA filters?
Okay, so HEPA and ULPA filters are two types of filters made of the same material. It's spun borosilicate glass. And the difference is really the density of the glass fibers and the size of particles that could pass through the fibers. HEPA filters usually filter out 99.99% of particles, 0.3 microns. ULPA filters filter out 99.999% of particles, around 0.12 microns. Really, though, for biosafety applications, for cell biology, HEPA filters are the standard, and ULPA filters really don't provide any extra protection. The main difference really is geographical preference. In Asia, ULPA filters are preferred just for geographical preference reasons. Most of the rest of the world, HEPA filters are preferred. And HEPA filters are the filters that are mandated in the standards like NSF or the European standard that govern biosafety cabinets.
Wonderful, thank you. All right, our next question that we have here is asking if you can kindly suggest which biosafety cabinet is required for fungal pathogens.
Great question. And for that, I'm going to refer you back to one of my introductory slides here, where we can look at the CDC's BMBL. It's hard to suggest a specific biosafety cabinet for fungal pathogens without knowing exactly which pathogen. But in this guide here, you can look up the exact pathogen. It'll tell you the types of containment and types of biosafety cabinet. And then you can work with your EH&S or biosafety officer and determine the exact type of biosafety cabinet you would need.
Wonderful, thank you. All right, our next question here. What could happen in a microbiology lab if the personnel did not correctly use the biosafety cabinet?
So in this case, there would be contamination of the samples on the inside, and there would be breach of containment to the lab. So if it was not used correctly, let's say if they had the sash open too high or too low, or didn't even use the blower and just used it as a dead air lab space, in this case, there would be not good things. So the users would be infected from whatever's on the inside, and whatever's on the inside would be contaminated from the lab. So we always say on the biosafety cabinet, wait for the airflow to stabilize. And Labconco biosafety cabinets have a very clear indicator of when the airflow is stable and safe. And always use the biosafety cabinet with the correct height sash. Never have the sash open too high or too low.
All right, wonderful, thank you. And it looks like we have time for one more question. This last question here is asking if you can provide a few more details about the types and amounts of chemicals that can be used in a type A2 biosafety cabinet.
Great, so I will show you this slide here. So when a type A2 biosafety cabinet is exhausted, and you can see the canopy on top, this is when the guidelines allow minute chemicals. So minute is kind of hard to define. Generally, though, this would be things like ethanol, isopropanol, phenol, chloroform, things that are used really commonly in cell biology experiments. And minute amounts, it's again hard to define, and it's really should be determined in consultation with your EH&S or biosafety officers based on the building's exhaust capabilities. But generally, the volumes used in standard microbiology or cell biology experiments on the orders of milliliters are generally safe.
Wonderful. Thank you. I do want to thank you again for your time today, Jeremy, and your important research. We would also like to thank LabRoots and our sponsor, Labconco, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.